Welcome to Recovering Evangelicals, a podcast for people who were once very comfortable in their Christian faith until the 21st century intruded and made it very hard to keep on believing. I'm Luke Jansen, and my co-host Boyd had a change of plans and won't be joining us for this interview. Today we'll be talking to Dr. Adam Rutherford, who has a PhD in genetics from University College London. Rather than pursue a career in research at a lab bench, he used his immense talent as an engaging science communicator towards speaking and writing through a wide variety of outlets. He started as a podcast editor and audio-video editor for the top-ranked science journal Nature, and has since become a frequent science presenter for several programs and documentaries on BBC Radio, and a frequent science journalist for The Guardian and many other newspapers and magazines. He's now back at UCL teaching university courses. He's written numerous books with provocative titles like A Brief History of Everyone Who Ever Lived, The Book of Humans, The Dark History and Troubling Present of Eugenics, and here's a good one, How to Argue with a Racist. He's been a science consultant in several major sci-fi films, including Ex Machina. The list goes on. He just loves to speak on science. And there's a long list of specific topics that he'll present on, including human evolution and ancient DNA. All of this makes him the perfect guest for our topic today about the origin and evolution of humans. So, Dr. Rutherford.
Hi.
Hello, sir.
How are you doing?
Thanks for joining me for this episode.
My pleasure. Good to talk to you.
Yeah, BBC have you running off your feet, but you're taking the time out of your busy schedule, so I really appreciate this.
Yeah, no problem at all. It's my passion project. This is what I've been writing about for the last 10 years or so, and it's really, the area that I find most exciting and a constant revolution in genetics.
Now, I've just told the listeners how you started in academia and then you caught the bug for educating the public, but maybe you can tell us why you took this less conventional trajectory.
I think that in very simple terms, it was because I was better at talking about it than doing it. And the lab is it's a competitive environment. I know a lot of people have a hard time during their PhDs and they come out of it sort of beaten and bruised, and the path into academia from there is very hard. Mm-hmm. That wasn't me. I had a great time in my PhD and finished it relatively tightly. I got on extremely well with my supervisor and did some work that I'm extremely proud of. We tried to get me on a postdoc in Canada, actually. It's at SickKids in Toronto, because that's where our collaborators were. But it didn't work out. And at that point, I thought, I'm going to switch over to the other side.
Our loss.
And I started working in academic publishing. So I was an editor at Nature for a few years. And then I just sort of branched out into being a more public-facing science communicator. The funny thing is that having gone from being a very narrow base, the genetics of eye development, to a very broad base, science communication across the whole spectrum, I've now gone fully back in the other direction, and I'm back at UCL teaching the history of genetics, working on population genetics, and so I've got a sort of a loop in my career. I'm back where I started.
Very good. Well, it looks like you've had a lot of fun doing that. So if it's okay with you, I want to dive right in because we've got a lot of ground to cover. Again, last week we talked to Dr. Donald Johanson about human evolution from an anthropologist's point of view. And near the end of that interview, he tantalized us with the prospect that the bones he'd been studying for decades are now being studied by geneticists who pull out DNA and give us a whole new perspective on human evolution. Who had the brilliant idea of trying to pull out DNA from an ancient bone? It sounds so counterintuitive because you'd expect organic material to degrade within days or weeks after death.
Yeah, that's exactly right. But it turns out, something that we're incredibly grateful for, that DNA is remarkably stable under certain conditions as a means of storing information. And so I think it's first touted as an idea in the 80s, but the technology was so far in the future at this point that I don't think anyone really pursued it. And then in the 1990s, in the late 90s, 97, I think it was the first paper published where some DNA had been pulled out of something that was already dead. It began to be a reality. Now, the problem with it is that although we say that DNA is stable enough to last we're now up to hundreds of thousands of years in the right conditions, it does degrade over that time. And over, the longer a time period is, the more bugs are going to eat it, animals, and bones get redistributed, and all just the environmental stuff that happens to organisms once they've died means that the DNA that you may extract out of an old bone it might not be the original DNA of the organism that had that bone inside them. So when we talk about recovering genomes from ancient hominids, from Neanderthals or Homo sapiens or whatever it is, we talk about getting the whole genome out. It's not really the whole genome. It's bits and pieces of the genome that we sort of staple together and come up with enough coverage that we feel that it's representative of an earlier species or an extinct organism.
Right. And these special conditions you referred to, I guess that would include, first of all, the DNA is inside this bone, which is a big, hard, calcified shell, and so it protects the DNA literally, physically. But then you also have to protect the DNA from water and from oxygen, I suppose. It has to be buried in mud to protect it from the oxygen. So these are some of the special conditions you're referring to.
All of those things. And this is why I think these types of conversations are really important, because it's become so much part of the scientific process that we can get ancient DNA, long dead people and long extinct species, that we've become a bit casual about the way we talk about it. It is still extremely specialist, extremely hard. And the DNA that you get out of these ancient bones is often in a pretty messed up condition and requires a lot of repair and statistical analysis and multiple runs and coverage in order to get a readable sequence. The bone you're referring to is the petrous. So there's this very hard, if your listeners feel behind their ear, there's a very hard bit of bone. And it's where a lot of the mechanics of our hearing are stored. It's the thickest part of bone and the hardest, most dense part of bone in the human skull. So it serves very well to protect DNA if it's been sitting in the ground for tens or hundreds of thousands of years. It's not the only source, though. The first Neanderthal that was sampled came from the humerus of a Neanderthal that was found.
The arm bone.
Yeah, exactly. Upper arm. And that was in 2009, I believe.
And Denisovans, I understand, was from a knuckle.
It was from the distal tip of the fifth finger of a teenage girl. So the two original specimens were the tip of the fifth finger and a big tooth, an unusually big tooth. And teeth are often really good sources of DNA because there's such thick enamel. And if you can get to the pulpy stuff in the middle, you might find some previously wet tissue there. But it was the finger bone that they got the DNA out of. But both of those examples talk to another aspect of the special conditions that preserve DNA, which is cold and dry.
Okay.
And so the DNA gets preserved in a much better condition the further north you go and the colder you go. And this poses a fundamental, deeply unfortunate problem for this whole field, which may be unresolvable, which is that, as Don Johansson would have been talking about in the last episode, the cradle of humankind, Homo sapiens is an African species, various areas of Africa, the Rift Valley and now Morocco, where we think that the earliest Homo sapiens specimens were from, are hot places. And all of those bones, I would be very surprised if we get meaningful DNA out of some of the older East African specimens.
Right, that's too bad. Now,
it is.
Speaking of the degradation of the DNA then, so DNA is very, very long. I mean, I've heard stories of when you stretch it out, it can be like a meter or two in length. That's the entire genome. But the DNA inside these bones are broken down into small snippets of just, so the genome would be in the millions and billions of bases. These snippets are hundreds of bases long, or a few hundred. And so I guess part of the problem is to, as you said, staple these things back together using computers and basically putting together a jigsaw puzzle.
Yeah, that's exactly right. And in fact, that is how the Human Genome Project works in itself in the 1990s. So the technology was, and the software, the computational biology was sort of set up for us to do this for ancient DNA about a decade earlier. We call it shotgun sequencing because one of the techniques that was developed was to basically smash up the whole genome into millions and millions of fragments. And then you use the software, powerful computers to align the bits so that you can work out a complete read. So the more times you read the genome, the more times you duplicate the content of the genome, the better the read. And then you get this sort of contiguous pattern and you draw from that the original sequence. So that was also the process by which we do ancient DNA. The problem, or one of the many problems, is that there may be huge gaps, there may be bits missing, there may be bits which have been degraded and are irretrievable. other genomes that have been in organisms chewing away at those bones for the last 100,000 years may have integrated their genomes into it. So you can't tell what's bacteria or fungus or other animals and what's human. Right. So it's a huge, you're right to say it's a jigsaw. I guess doing a live human genome is a jigsaw where at least all the pieces are.
Right.
Doing dead humans, you're doing a jigsaw where you may have multiple boxes of different jigsaws, and loads of the pieces are actually missing.
Right.
So you're trying to just get a broad picture of roughly what it looks like.
Right. And to give the listeners a bit of an image in their minds, I'm sure they've seen, I've seen a few movies where people get their hands on government documents that have been shredded, and they very carefully paste them all back together again, lining them up and pasting them. So this is a similar kind of a thing using computers.
Yeah. I think that's a good analogy, actually. So I think I'm thinking of Argo is a film where they do that, which has a great Canadian theme in it as well. Yeah. It's that times a billion, right?
Only, which is why they use supercomputers then, obviously.
Exactly. None of this could be possible without the advances made in computational biology over the last 20 years. You think about it sometimes on films or on TV programs like CSI, when they have the geneticists and they hold an X-ray up and it's got little bars on it, right? And they look at the DNA sequence and say, oh, this was the murder or whatever. Well, there's a couple of things about that. One is that technology. We stopped using that in about 1995. So when you still see it on TV today, we don't really do it like that. But the second thing is...
And just so, sorry to interrupt you, but just so the listeners know what you're saying, when you say hold up an X-ray, you don't mean an X-ray of the person's skeleton, that kind of an X-ray. This is an X-ray of a genetic breakdown of the material inside of a cell.
Exactly. So it's got like these ladders running up them, and it's where you can see the individual letters of the genetic code, and they have a big revelation because they suddenly see that there's a mutation there or whatever. When they do that, they're looking at like 50 letters of genetic code, which is, most genes are about, round about, the average is about 1,000 letters. And some of them go up to tens of thousands. And the whole genome is 3 billion letters of genetic code. So in one of my books, I described it as being, it's not a needle in a haystack. It's trying to find a broken needle in thousands of fields of corn, none of which have been vaguely tended to for the last billion years.
I could see that would make it very difficult. But now, so using this technique then, how far back has science been able to go?
Well, it's a great question. And I think in one way, the answer is all the way, because and when I say all the way, I mean to the last universal common ancestor of all life on Earth, which is a sort of hypothetical organism that we call LUCA, last universal common ancestor.
Right.
And by looking at mutation rates in genes and looking at shared genes between individuals, between populations, and between different species, we have a sort of molecular clock, and we can see where genes have, where species have split from each other because two different species have similar genes, but which means that they had a common ancestor, which of course is the central Darwinian paradigm. So we can wind that all the way back, and we now think that, off the top of my head, I think the number was 355 genes that are at the core, the base of all living organisms that have ever existed since LUCA. And that's an inference. It's an inference based on how we understand how DNA is passed from cell to cell, from generation to generation. It's pretty accurate up until about sort of 2.5 billion years ago, when complex life, eukaryotic life emerged, because we have a pretty straightforward tree from that point on. Before that, there were two domains of life, which are archaea and bacteria. And they look so similar to each other down a microscope that the archaea weren't even distinguished from bacteria until the 1980s. But they're actually, very, very distinct in many other biochemical ways. But archaea and bacteria do something which eukaryotic life doesn't do, which is that they can swap genes between siblings rather than from parent to child, as it were, in bacteria. And what that means is that there's a sort of event horizon of how far back we can see using molecular clocks of genetics, because they don't just bifurcate in a sort of branching way. they share their
Right.
genes over
Exactly.
complex networks.
Okay. So that's genetics in general, and I want to come back to that in a moment, but we left off with the one question: how far back in time have we been able to go using this technique with the bones, getting DNA from the bones? A related question then was, how much of the hominid family tree have we been able to explore using this new technique?
Yeah, well, that's an ever-moving target question, and the answer is, well, it's a complex answer because it's clarified a great deal of questions from our relatively recent past, so the last 100,000 years. And it's also revealed quite how little we know about human evolution. So I'm 47, right? So I grew up with Lucy being, I think Lucy was 1975.
74,
right?
I think it was, yeah.
Okay, so I was born in 75. So Lucy has been in my life my whole life.
Right.
And I was a very nerdy kid and super into human evolution from a very early age. So I dug up some of my old books that had the human story in them. And they've got these very definite maps, branching trees of how we got from Lucy to us. And they're clear, right? We knew that story then. With every new discovery, they just fitted in. There's Australopithecus afarensis, and there's Homo erectus and various branches, and Homo heidelbergensis, and then Homo neanderthalensis, and then eventually Homo sapiens, right? And we knew it. And even when I was at university, so this is in the 90s now, and I did paleoanthropology, there was a great deal of confidence in those pathways. Ancient DNA, the ability to pull out the actual genetic code from our ancestors, just tore all of those things apart.
Wow.
It meant we just didn't. I don't think there are any direct lines we can draw with great confidence outside of the ones that we know happened because of DNA anymore. And maybe Don Johansson would disagree with me, and other paleoanthropologists would say, well, we do know that, but the hard evidence is now that we've got at best dotted lines between the things that we once thought were definitely our ancestors.
Right. And that's one of the good things about science, that when new data come in, we scientists are willing to revise the story according to the data we have at hand. So
absolutely.
So let's now get to the one important question I really wanted to hear your thoughts on. Long before scientists learned to get the DNA from ancient bones, we already had quite a bit of evidence that humans had an ancestor in common with the other primates. So things like the chimpanzees and gorillas and orangutans. What were some of those clues that we had a common ancestor with them? And maybe start with the one that's always intrigued me, the story of the human chromosome 2.
Yeah, absolutely. So the whole idea of common descent was, first first described really accurately by Darwin, and was based on morphological characteristics. So the fact that, we have the same bones in our hands as whales do in their fins, as bats do in their wings, and so on. So those were the setups for common descent. And when there was lots of debates over the late 20th century with people who denied evolution, particularly biblical creationists, these were some of the standard tools that would wheel out and say, look, the evidence is unequivocal here. And then when genetics entered the party, it just became even more unequivocal. So
exactly.
It's just reinforced at a molecular level that those morphological similarities, DNA was saying exactly the same thing, but with even more accuracy and precision. And we mentioned it a minute ago when we were talking about how if we look at the fact that there are genes that are shared between closely related species and distantly related species, and they're very similar, or we can tell how much they've changed over evolutionary time by the mutation rate of the individual letters of DNA. What it shows unequivocally is that there is shared ancestry, right? And so that common descent and descent with modification, that Darwinian phrase, is unequivocally true. Most changes that happen at a genetic level are small. They're individual letters that change over generational time and introduce a slightly different shaped protein, which might be selectable or deselectable. But then there's also these big scale macro changes that happen in our DNA. And they are much rarer because they tend to produce big changes. Chromosomal level changes tend to produce non-viable babies. And we know about examples of that they're called translocations. So big translocations in human embryos often result in lethality. And the best known of these is Down syndrome. So people with Down syndrome have an extra chromosome 21 and have a range of disabilities as a result. But mostly these types of big structural changes in chromosomes result in non-viability. But they do happen. And the one you're alluding to is one of the great stories in evolutionary biology, which is that when we count the number of chromosomes that gorillas and chimpanzees have, it's 48. So 24 pairs of chromosomes. And we have 46, 23 pairs of chromosomes. So you think, well, when you line them up, almost all of them match pretty closely. You can see that this human chromosome is basically the same as this gorilla chromosome. But then there's this weird number crunch that they've got more than us. And what we see unequivocally, again, very elegantly demonstrated, is that our chromosome 2, which is huge, is a fusion of two of the gorilla and chimpanzee chromosomes.
Joined head to head like Siamese twins.
We can tell that a number of ways. So not just where the genes are on both of the great ape chromosomes and where they are on the human chromosome 2, but also there's these sort of markers on chromosomes. There's a thing called the centromere, which is a little pinched bit in the middle. So when people see those pictures of chromosomes, they sort of look like, I describe them as being like socks, right? They look like socks that have been twisted in the middle. The middle bit is called the centromere, and the two gorilla chromosomes have two centromeres, and our chromosome 2 has one big centromere, but it also has the remnants of two other centromeres, which are in the same position as they are in the gorilla and the chimpanzee chromosomes. So what we think happened is at some point within the last 11 million years is the conservative estimate, but maybe more like 6 or 7 million years, where the branch that becomes Homo, Australopithecus, and then Homo, we presume, and the branch that goes towards gorillas, chimpanzees, bonobos, and orangutans, that there was this massive fusion at some point when egg and sperm were being formed in an ancestor common to all of the great apes. Instead of those two chromosomes in that ancestor remaining separate, they smushed together, and it didn't cause that fetus to die. It resulted in a new lineage, right. And that's us.
Yeah. And I recommend the listeners look at a picture of this. Just type in human chromosome number 2 and maybe read a short story around that. A picture tells 1,000 words, and it's undeniable. But there's other lines of evidence, broken genes, pseudogenes, and proviral sequences. Do you want to say anything about those?
Yeah, sure. Absolutely. So one of the funny things about the human genome that was revealed in the Human Genome Project 22 years ago, when the first draft sequence was released, was something that I think that a lot of us hadn't anticipated. So we mentioned that the whole human genome is like 3 billion base pairs long, 3 billion letters of DNA. So it's huge. But we found back in the Human Genome Project days that less than 3% of that is actually genes. It's actually the stuff that makes proteins, that does stuff in our bodies. So the vast majority of all of those chromosomes in that picture that you just looked up is not genes. So what is it? Well, large chunks of it are sort of structural things to actually hold them in the right shape. Things like the centromere that we talked about for chromosome 2, they are scaffolding to hold the DNA in the right sort of formation. There's bits that sit at the end of chromosomes called telomeres that you might have heard of. And they sort of cap the end of DNA to stop it fraying, like the little plastic bit at the end of laces. Mm-hmm. And then there's huge bits which do nothing, we think. There's huge bits which are regulatory sequence, turning genes on and off. And then I can't remember the exact percentage now, but it's like 0.5% or 1% is stuff that used to be genes. And we can see that they used to encode proteins, which is what genes do, but they've just rusted. Right? They've just mutated, and we can see the remnants of them. They're fossils, right? They're fossils of genes that used to do stuff in our bodies, and we stopped using them. And, evolution is a perfect example of use it or lose it. If you're producing proteins that don't do anything, then you're wasting energy. Mm-hmm. The best example, the one that I like, the one that I teach, because I think it's so elegant, is
vitamin C?
No, but that is a good example. I was going to go for olfaction, so smell.
Okay, right, yeah.
So, when we talk about how dogs and mice have much far greater senses of smell than us, and I think the estimate is there's something like 900 genes in the average dog genome for detecting specific smells. We have about 450, between 4 and 500. But if we count all of the pseudogenes, so the things that used to be genes, and we compare them to the olfaction genes in dogs, they're all accounted for. Mm-hmm. All of the same genes are there, but we just haven't used them for millions of years. And so they don't get cut out of the genome. They just sit there, mm-hmm. and they rust out of function. So we are worse smellers than dogs, but we carry the fossils of having olfaction in the same way that dogs and mice and whales do.
Right. And I don't think you mentioned, or you didn't highlight this, but it's not just that those genes which are not working anymore, that they've rusted, as you say, but they're rusted in exactly the same spot in the human and chimpanzee genome versus other animals that have lost the same gene.
Exactly. There's another version of this story which is done in the opposite direction. So my PhD was on the eye. It was on eye development. It's about how the retina, how we turn light into perception and vision. And instead of talking about the genes that we've lost, in the human evolution story or in the primate story, there's exactly the same version in reverse, which is that many monkeys today, old world monkeys, are dichromatic. So they can only see in 2 primary colors. And we are trichromatic, which means that we see in 3 primary colors, the primary colors of red, blue, and green. What that means at a genetic level is that there are 3 different types of molecules which are called opsins. And the opsins sit at the back of the retina in our rods and cones, mostly in our cones, and they are tuned to pick up specific wavelengths of light that correspond to red, blue, and green. And those are the primary colors. So there are 3 genes that encode those 3 opsins, red, blue, and green, or actually short, medium, and long wavelength is what we actually call them. Now, our ancestors only had 2 of those. And what we can see exactly when one of those genes was duplicated in one of our lineages, so that when a cell was reproducing, instead of just copying one of them, it accidentally duplicated one of those color genes onto the X chromosome, actually on both of them on the X chromosome. And what that meant was, from a sort of functional point of view, is now you've got 2 genes which are doing the same thing. They're both encoding perception of blue light. And that's a wonderful template for evolution to bounce off of, because what it means is you can muck around with this one and you still have blue perception. Right. So if this one mutates over a few generations and becomes attuned to green or red, then all of a sudden, and when I say all of a sudden, I mean over thousands, possibly hundreds of thousands of years. Right. That's the time frame we talk about. Right. But all of a sudden, you've got 3 versions of color perception genes. And if that's advantageous to the organisms that have them, possibly because we could identify colored fruit more easily, or that's one of the reasons that's been suggested, although we can't ever really demonstrate that. But what it means is we can tell that one organism in our ancestral tree has 2 versions of it, and we got 3, and we can tell when it happened, and we've acquired information through mutation.
Right. Now, let's get to a more salacious part of the story. Dr. Johansen hinted at the idea that we humans have been having sex with our genetic cousins. And because of that, many people now have a certain amount of Neanderthal DNA themselves, even today.
Well, undeniably true. Undeniably true.
It's something like 3 to 5%, I understand, at least if you're non-African, as I understand it, because, well, maybe you know more of the story behind this, why some of us have 3 to 5% of Neanderthal DNA.
Yeah. So Neanderthals were discovered in the first couple of decades of the 19th century. And for all of the next 100 years, 90 years, I suppose, they have been designated a different species because they're morphologically different, right? So they've got broader noses, both externally and internally, so in the sinuses areas, a heavier brow, larger cranial capacity, so bigger brains than us, a big barrel chest, right? And for that reason, Neanderthals were designated as a separate species, Homo neanderthalensis. In the 60s until the end of the 20th century, it was a big debate about what our relationship was with the Neanderthals, because we overlapped in space and time by about 5 to 10,000 years, mostly in Europe or Eurasia. So whilst it was emerging as a result of the work of people like Don Johansen that Homo sapiens is an African species that then moved out of Africa and populated the world. The idea was that we had a common ancestor with Neanderthals maybe 600,000 years ago in Africa, and an earlier branch had migrated out of Africa and had resulted in the Neanderthals and them being a sort of Eurasian species. But then within the last 50,000 years, Homo sapiens comes out of Africa and meets up with Neanderthals. So they overlap in time and space. Mm-hmm. And then the question was, well, what happened, right? What happened when Homo sapiens meets Homo neanderthalensis, because they don't exist and we do. And so there was lots of debate about, why one survived and why one didn't. Were we the same species? Most people said no, absolutely not. Did we eat them? Did we have sex with them? What was the relationship like? And there was a lot of speculation about that. Now, in 2009, in such a landmark paper, because this is why we're having this conversation, it just ended that question overnight with the work led by a guy called Svante Pääbo, who's based in Germany, who's a real pioneer in this field. But he and his team got DNA out of the humerus bone, so the upper arm bone of one of the prototype specimen of Neanderthals that had been found 100 years earlier. And what it unequivocally showed is that it had Homo sapiens DNA in it. And so what we found is that we've now got the Neanderthal genome, and it's got Homo sapiens DNA in it. And now that we've got Neanderthal DNA, we can now look at our own DNA and compare it to it. And as you say, all people, or almost all people descended from largely European populations, have a significant amount of Neanderthal DNA sitting in them. When you do a 23andMe or an Ancestry.com test, one of those commercial ones, the number can creep up towards between 3 and 5. I think the real estimate based on the academic work is more like 1 to 2.
Okay.
So there's a slight sampling bias that happens in those commercial tests. And so it answered the question, not only we did overlap in time and space with them, and we definitely had sex with them.
Yeah.
Right. So there's an interesting language thing here, which I had been using the same language as you until relatively recently. And then someone pointed something out to me, which made me laugh and made me change the way I talk about this. You don't talk about your grandfather as being someone who had sex with your grandmother, right? Your grandfather was your ancestor.
TMI.
Okay, yeah, I'm sorry. But do you know what I mean?
Yes.
So when we talk about Neanderthals as being our cousins, they weren't our cousins. They were our ancestors.
Right.
If you carried DNA of Neanderthals in you, they were our ancestors. So they weren't our evolutionary cousins at all. They were our ancestors.
Right,
they were our great-great-great-great-grandparents just as much as the Homo sapiens was. So in a sense, I tend towards the slightly revisionist version of Neanderthals, which is that they were us. They did look different from us a little bit, but not so much that your and I, great-great-great-great-great-great-great-grandparents, got it on with them. And so I think that it's important that we sort of recognize that they weren't different. Well, they weren't significantly different enough for us to warrant calling us cousins. They were us.
Really. And I've also heard the word subspecies, which I don't think that changes your point. And Dr. Johansson was talking about hybrids and how you can get speciation out of hybrids. You bring two species together and have a hybrid. So I don't know if these are terms that are relevant in this context, but very interesting things.
Well, they are kind of, and I'm a bit. So paleoanthropology and the more traditional routes where species are delineated by physical differences, so the big brow ridge or the barrel chest, and you accumulate enough of them and then say, I think these things are different enough from this species that we can call them a different species. Mm-hmm. And that's taxonomy. That was invented by Linnaeus in the 18th century, and it is how we describe all living things. We give things a genus, which for us is Homo, the humans, and then you give it a species designation, which for us is sapiens. For Neanderthals, it's Neanderthalensis. For, I don't know, pick one, Homo floresiensis, the Hobbit man, Homo erectus. They're different enough that they warrant being in the same genus, but a different species. Mm-hmm. Now, I am intellectually hostile towards taxonomy because I think that if it works, it can be useful. But where it doesn't, it becomes very destructive and not descriptive, because the species are defined by being, there are many species definitions, but the most commonly used one is two organisms that cannot produce fertile offspring. So they can have sex and they can reproduce, but the offspring are not fertile themselves.
Mm-hmm.
So they won't continue their DNA. That won't be passed down into the future. Well, that's the standard definition. That's what we teach school kids. That's what we teach first-year undergraduates. But what we teach second-year undergraduates is if Homo neanderthalensis and Homo sapiens were different species, then why do me and you have 2% of Neanderthal DNA? Either they weren't separate species or the species definition is wrong.
Right.
You can't have it both ways.
Exactly. And the classic example that I've heard that argues we have the definition wrong would be, for example, a Great Dane and a Chihuahua. Two dogs, same species. The Great Dane is a giant animal, a small horse, in fact, and the Chihuahua can fit in a teacup, a coffee mug. They're not going to be reproducing, and it's just not going to happen without, at the very least, a stepladder, and yet they're still the same species.
Yeah, that's right. And there are physical mechanisms which prevent reproduction, such as that. What your listeners don't need to imagine that. And then there's genetic ones as well.
Right.
So, for example, we think that one of the rules of successful reproduction is that you have to have the same number of chromosomes, because there's a particular process that happens during reproduction, which is the chromosomes have to line up. So you have to have the same number of chromosomes in order for the offspring to survive. That appears to be a fairly hard and fast rule. But the truth of the matter is that the rules that we apply in biology always tend to fall apart with either exceptions. They're more like guidelines. We've already talked about how the fact that there was a point in time in the last 10 million years or so when one species produced an organism that had a different number of chromosomes to it, and now that's us.
Yeah.
We also know that equids, so the broader horse family, seem to be able to reproduce in between different types of horses with wildly different number of chromosomes, and we don't know why, right? There isn't an explanation.
Yes, I get that. We're getting short on time. One last scientific question, and then we might get into some more socio-political issues. But the scientific one, it's a bit more technical. I hope you can weigh in on this. And I want to get into this because it's going to be foundational for some important questions we'll get into later, not in this episode, but in future episodes. And this is the idea of tracing the maternal or mother's lineage back for thousands of years separately from the paternal or father's lineage. And so geneticists have this really cool trick to separate the genes which could come only from the mother and genes that come only from the father. Could you tell us how they do that?
Absolutely. So almost all of your DNA is inherited from both parents. So you get one set of chromosomes from your mother and one set of chromosomes from your father. We've already mentioned we have 46, so it's 23 pairs. But it's actually only 22 pairs because there's the sex chromosomes, and women have two X's, which means they get an X from their mother and an X from their father. And what that means is that the egg has one X in it, and the sperm that arrives that fertilizes it contains an X as well. So the fertilized egg has two X's, and that becomes female. And if that sperm contains a Y chromosome, then that individual is male, right? What that means is that the Y chromosome describes a lineage that can only be traced through the paternal line. Y chromosomes are only ever inherited from fathers to sons. And it means that if you look at the genes on the Y chromosome, you have effectively a pathway, a trajectory, which traces back a male line only.
And that can go back thousands of years, that paternal lineage.
Thousands of years.
Right.
Yeah, all the way. It doesn't recombine, so
Right.
It doesn't shuffle. The Y chromosome doesn't shuffle in the same way that the other chromosomes do. And for the maternal line, there's a similar trick, but it's slightly different, which is that there is a separate DNA called the mitochondrial DNA. Mitochondria, these little subcellular units which are responsible for generating power in our cells. So they're incredibly important.
We've talked about these before with Dr. Shapiro, when he mentioned one kind of bacterium, a cyanobacterium, swallowing up another bacterium, one of the archaea. And that became a subcellular machine that produced energy molecules for the host bacterium.
But they are outside of the nucleus, and all the DNA is kept in the nucleus apart from mitochondria, which have their own genome. They have their own tiny little ring of DNA, and that sits in the egg cytoplasm, so the jelly-like stuff that is separate from the nucleus. And what that means is that it's only passed down maternally. So the mitochondria is passed from mother to both children, regardless of what sex they are. So if you look at just the mitochondrial DNA, you have a maternal lineage. You can see a pathway that goes from mother to, from children to mother all the way back. So you've got these two edges, or these two lineages that allow us to trace back specific lineages which are sex dependent. Now, a lot has been made of this over the years. The first ancestry tests and the first genetic ancestry and genetic genealogy were really only based on mitochondria and Y chromosome lineages because they were the easiest ones for us to identify. And they became kind of fetishized in a sense in those early years, the first few years of the 21st century. And concepts like mitochondrial Eve and Y chromosome Adam became very popular ideas that you could trace back your lineage all the way to a foundational female from whom all humans are descended. And similarly, you could do that with the Y chromosome. They got a catchy name, which was mitochondrial Adam and Eve. Those two, where they came from, were separated by hundreds of thousands of years. So it doesn't reinforce the biblical story at all. But those sort of catchy memes, those sticky titles become very popular. And I think they've done us a disservice because they represent a very, very small proportion of our total amount of DNA. Less than 2% in total is the Y chromosome, which is a very tiny, stunted chromosome. No comment on the sort of social implications of what I just said. It's got only a handful of genes on it. It's got like 70 genes on it, and most of them don't do anything. But they do determine maleness. And the mitochondrial genome is incredibly important. I think it has 37 genes on it, something like that. Someone will write in and correct me. But they represent a very small proportion of the total amount of genetic information in a human being. So when we trace those lineages, we are addressing the fact that we have these traceable lineages, but we're ignoring the rest of your DNA, the vast majority of your DNA, which is inherited from both parents. So it became a very linear story. And I think the public got hold of it as a sort of really easy to grasp, simplified version of actually a very complex picture, because really we're talking about populations. And we're not really talking about individual people. We're talking about huge populations that interbreed and from which other populations are derived, and they mix and mate and fight and do all the things that humans do. The real story of human evolution is that we are good at two things: moving and sex. And as a result, human history is been an incredibly messy, those maps that we draw where you have big arrows that say we went out of Africa. Some people went east towards what we now call the East, and some people went west towards Europe and Britain. And the people who went east crossed the Bering Land Bridge and then populated the Americas, and the indigenous people of the Americas are descended from that. All those things are very broadly true over geological time frames. But the real story is, what we do, which is we get born, we meet people, we fall in love, we move to the other side of town, or occasionally we move to a different town. And in the modern era, we might move across the other side of the country or even to a different country. But I think it's important for us to think about human evolution, not just in those geological time periods, but also in lived lives. We talk about out of Africa as an event. Well, people have calculated the rate at which the migration out of Africa happened. And it's like, 5 miles every 1,000 years. It's not an event. It's not migration. People fall over faster than that. So we have to zoom out and zoom in. And I think that mitochondrial Eve and Y chromosome Adam story, it gave a much too sort of linear, simplified version of actually what is, these are people. Their lives were as complex and messy and driven by sex and competition and movement, migration, and what was available as our lives are.
Okay. Now we've got just a few minutes left. I don't want to keep you from your lunch, but I do want to get into to this more socio-political issue. You present in front of a wide variety of audiences, mostly the general public who have little training in science. And sometimes the science you're presenting goes against their worldview, especially religious worldviews. And in some cases, you'll actually take a direct stand against those worldviews. So, for example, you'll speak against groups who are in one way or another anti-science or who promote pseudoscience. Things like, for example, the link between autism and vaccines, You've written and spoken on things like alternative medicines and homeopathy, parapsychology, those sorts of things. And at times you've taken direct aim at religious groups. I was intrigued when I looked at your biography and found you've written articles that criticize the teaching of creationism of science. A very interesting article, or actually a 10-part series on your experience with the Alpha Course. And you were one of 42 authors who compiled that Atheist Guide to Christmas.
Well, we were talking about evangelical and creationism, and I think that I don't regret or withdraw anything I've said in the past, but I do think there was a youthful vigor about some of the things that I've written. And it's not that I don't think those things anymore. It's just I think it's more about picking one's battles. I actually come from a Catholic family and have a lot of time for religion and go to church with my mum, several times a year. There was an interesting point that happened a couple of years. Well, I say a couple of years ago. I mean a couple of years ago before COVID times, when I was at Mass in our church, and the father leader, who's been my vicar for the whole of my life, introduced a new prayer. So we always used to pray for the sick of the parish and pray for various things very specifically. And then one year he said, and we pray for the outlawing of stem cell research. And I thought, wow, man, that's really specific. I don't know whether he was, I was 4 rows back. And I thought, are you looking at me when you said that? Because I used to do stem cell research. And I just thought, damn, that's a real problem for me, because, the sick of the parish might not be so sick if we continue doing some stem cell research. But, what do you do in situations like that? I'm not going to stand up and argue with Father Leader. But I think it didn't help me maintain my sympathy or my sort of cultural sympathy for the culture that I have been born into. Another thing that may be worth mentioning is that I'm the new president of the Humanist UK, so the Humanist Association in the UK. I take that role on in this March. Took over from Alice Roberts, who some of you might have heard of. Right. Very wonderful science communicator and a good friend of mine. And it's a 3-year position, and I think I consider myself a humanist, which is non-secular and not religious, but believes in celebrating humankind and celebrating our achievements and recognizing science and objective reality. And it's not antagonistic towards religion, but it is antagonistic towards religious oppression.
Exactly.
And those two things are different.
Absolutely.
So as I hit my 50s, I'm taking a stand, but in a more sophisticated way.
Good. Today we've been talking about hominid evolution, and it goes without saying that will catch the attention of religious groups. Have you had any interesting interactions with them?
These are valid questions, and I've written and said all those things, and I think I don't withdraw any of them. I have calmed down. I think as I've got older, I think you pick your battles with less youthful vigor. And there was a period that many of your listeners will sort of remember towards the end of the 20th century and the beginning of the 21st century, when this became a sort of hot button topic. And I'm not excusing myself here, but just to give some context, this became a way to talk about science in public and talk about evolution in public. And in the UK, it has largely vanished as a conversation now. I don't want to claim victory, because I don't think we should think about things as sides. But we have a national curriculum which removed the teaching of creationism as part of science, and so it became a non-issue there. There are still religious groups that are wedded to it, but they're very fringe and non-vocal. And it stopped being part of the popular discourse after just a few years. I don't think that is true in America. I think that conversation has not changed in the same way. And
not at all.
And maybe it's the same in Canada too. I don't know.
Well, no, we're closer to you. I'd say largely we're much like the British model, but certainly in America, the US, this is a very hot button issue.
Yeah, it hasn't gone away at all. I became, this is a terrible thing to say, but I think I became bored. And, it was a platform that helped launch my career. I started writing in the popular press in 2008, 2009, and that was the Darwin centenary, 200 years since he was born and 150 years since the publication of The Origin of Species. And it was a big deal over here. There's lots of celebrations, tons of events. And if you're involved in evolution and evolutionary genetics and do public facing science, it was wonderful. We were doing tons of documentaries and writing, and it was amazing. And I think that I got to a point where I think I'd said everything I needed to say about it, and that other people could continue to have that conversation. And what I say, I don't mean to sound patronizing, but to people who follow, but it does appear to be a fixation of young scientists who are up for scraps. They like a Barney because there's always rich fodder there, and important battles to be fought as well. When it comes to challenging other forms of pseudoscience, well, I think I've refined my views on this, because there are some battles worth having, and there are others which it's worth walking away from and doing something more interesting, or making a cup of tea. And we've gone through an interesting period in the last 2 years with regards to that, because there has been a colossal amount of scientific misinformation or misappropriation. In some ways, it's been terrible, because there's been so much misinformation out there and filtering, terrible stats, terrible misunderstanding of science or how science works, whether masks work or not, whether vaccines work or not. There's been a lot of battles in there. In another sense, I think it's been very positive for science, because all of my work throughout my career has been not about telling people facts or telling people truths, but telling people how science works and why science is a way of knowing, right? It's not a bank of facts. It's how we find things out. All of the methodologies that we've developed over the last several hundred years are there to test reality, right? And to remove our prejudices, our built-in psychological biases from our view of understanding what is real. And it's an ever-moving target. Science is a dynamic thing, that all results are conditional and transitional. Right. The ones that last are the ones that are tested and they don't fail, like evolution by natural selection or gravity. And I think that COVID has done something. I hope the analysis of this will go on for many years, and people will be writing theses about this for centuries. I would like to think that the scientific process has been exposed to the public in a way that it could never have done in any other scenario, because they were seeing how we gather data, how we develop ideas, how we publish things, how they turn out to be wrong, how we assess data, how statistics can be applied and misapplied in understanding specific bits. And also, one of the greatest things that has happened in the 21st century, which is from zero to vaccine to delivery of vaccine to the majority of the population in less than 18 months, which is, that should be marked as one of humanity's greatest achievements.
Absolutely. And one last comment on that. I understand that the technology that came up with this vaccine, that produced the vaccines, can now be, they're talking about applying this against cancers and a wide range of diseases. It's a breakthrough in medicine.
Absolutely true. I mean, vaccines have been around since, well, in a formal sense, since the 19th century, but, there's legends of people in China, like, inhaling crushed up scabs and things, and they had immune responses. So that's been around for a very long time. But the new techniques and the speed at which we could develop these different types of vaccines, one of which is brand new, using RNA, so rather than DNA, the sort of a messenger version of RNA, have all been enabled by technology that was invented during the Human Genome Project years. Right. There isn't a vaccine available now that isn't the result of genetic engineering that was invented in the last 20 years by studying genetics and inventing new tools. Insulin, another example of something which, millions of people all over the world need to live. And it used to be grown in pigs and filtered and grown in vats, and we took it. Now it's all grown in, it's all genetically engineered, and people don't even realize it. Right. It's changed the world. Absolutely. It's changed everybody's lives.
Yeah, absolutely. Now, one last thing. Several times in our episodes, we've seen a very interesting phenomenon of experts in a given field having to very carefully mince their words out of a concern about how that could be used by, in air quotes, the other side. We saw that, as you just mentioned, the misinformation around COVID. We saw that in Fred Hoyle being resistant at first to Stephen Hawking's idea of the Big Bang. And we saw it when Dr. James Shapiro and Jeffrey Schloss told us about the Royal Society meeting in 2016 when a large number of scientists wanted to update evolution theory with the very latest ideas in genetics. I'm wondering if you might have been at the Royal Society meeting, or at the very least you're quite dialed into the things around the Royal Society meeting. So what's your take on that meeting in particular and this idea in general of experts being so cautious about how the public will take their ideas out of context?
Yeah, well, I mean, there's a lot of things going on in there to try and address that question, which I think is a really important one. I wasn't at that meeting, although I go to the Royal Society quite often, and I remember it happening. What I do think is that it's a relatively minor academic dispute about things that are fundamentally not, the two camps weren't really disagreeing with each other in any sort of substantive form. And this is how academia works. It's how science works. And I guess that the protagonists in those debates, I'm sure that, they're all good scientists, and the people at that meeting, I think I've got a great deal of respect for. They were billing it as a sort of reassessment of the modern synthesis, which was this sort of fusion of genetics with Darwinian ideas that happened in the 1920s and 30s. And sometimes, you draw attention by saying we're tipping everything out, we're upturning Darwin. In 2009, we mentioned how that was the anniversary. New Scientist, one of the sort of cornerstones of the public-facing science communication, their front cover on Darwin's anniversary was Darwin was wrong. And the subtitle was like, upending the tree of life. And I was reading that thinking, well, that's bold. And it was actually it was an article about another facet of genetics, which is massively overhyped, but is really interesting and immature. And that's epigenetics and transgenerational epigenetics, and whether that did have a serious challenge to our views of natural selection, to which the answer is no, an absolute unequivocal no. But sells a lot of copies. If you can push the big guy off a pedestal, then you're going to be the most famous scientist in the world. Now, that Royal Society meeting wasn't one of those. It was some interesting incremental work. But it's interesting that you bring it up, because it shows something about how science works. Always, science needs to be contested. Right. Nothing is truth in science. When you publish a big paper or any paper, that isn't a mark of it being true. It's a mark of it being worthy of further discussion in the field. Right. And that, again, I think is a misapprehension or misunderstanding that the public often don't recognize exactly what that scientific process is. Right. We've come up with some results, and now we're going to defend them. But I say this often, the best thing that a scientist can do is recognize when they're wrong and change their views. Exactly. So we set out to be wrong. Right. The goal of research is to be wrong. Okay, Adam, we've gone far over time, and you've really indulged us here. I'm so thankful that you did. Before we go, do you want to tell us anything about some new directions you might be taking? Oh, sure. Well, thanks, man. It was great fun talking to you as well. Some of my favorite subjects, as you could probably tell as I waffled on. I suppose if I was going to plug my pluggables, I've just published a book in this country called Control, which is about the history and the legacy of eugenics. And that's coming out in the States and Canada in, I think, September or October. It's got the same title, but we're just finishing off I've rewritten it for the North American market so that it's got fewer references to British politics and more references to the eugenics that was enacted in both Canada and the United States. And it's a very personal book for me, because I come from the birthplace of eugenics. I was in the lab that was founded by Francis Galton, Darwin's half cousin, who invented the words and developed the whole field. And that is the department that I'm a lecturer in now, where I teach the history of eugenics. So that'll be my that's my next big book, which comes out later in the year. Okay, great. And I'm going to put links to your homepage on our show notes. Thank you very much. Adam, I really appreciate this. Not at all. It's been fun. Okay. It's been fun. I am hungry now, though. Okay, thanks. And all the best in your exciting career. Thanks, Luke. And if you want to talk again, just let me know. Sure. Good weekend. Bye now. All right. Cheers, man. You've been listening to Recovering Evangelicals with Luke Jansen. Send your comments, questions, and suggestions for new episodes to lukejjansen@gmail.com. Join our private discussion group on Facebook by searching for the Recovering Evangelicals podcast group. Look for the logo that you always see at your favorite podcast catcher and ask to be let in. Subscribe to lukejjansen.wordpress.com if you want to get advanced releases and group posts, or browse through our archive for a full listing of all our previous episodes. And please help this podcast by sharing with a friend and leaving a review rating at your favorite podcast catcher.